Okay, today we'll do a brief overview of fungi. So what are fungi? These are a group of heterotrophic, so they eat. They don't produce their own energy through photosynthesis. They actually have to eat things to get energy. They're heterotrophic eukaryotes that include organisms like molds, yeasts, and mushrooms. And here's just some pictures of the various diversity of different fungi. And fungi, most of the time, they're what we call saprozoic or saprophytic, meaning they consume dead organic matter. So you can think of them as decomposers that help recycle nutrients into the soil and just for other organisms. And there's very large variety of them, but overall they can be unicellular. Usually asexual reproducers, which are known as yeast, or they can be multicellular and sexual reproducers, which are generally known as molds. These are just general classifications. And I will say there's caveats to this, of course. So there can be asexual reproducers, there can be unicellular forms that are also sexual reproducers. Okay, so let's look at some of the features. Unicellular fungi, like I said, usually reproduce asexually through what we call budding. So budding, it's similar to mitosis, where two cells replicate their DNA and then divide, right? But instead, what we have is that we have this larger cell kind of pinching off and forming what we call a bud, which is just a little beginning of a small cell. And then the cytoplasm and, of course, the cell wall will come and split the two cells together, but they'll kind of still be attached like this. And then as the smaller cell grows in size, it'll start to detach and then come off. You can see how one bud can give rise to multiple other buds, and then this can keep going into a chain. And in contrast, multicellular fungi, they form these long filament-like structures called hyphae. And hyphae, they're kind of like, you can think of them as like roots. So if you look like under a mushroom in the soil, you'll see this network of hyphae that kind of look like plant roots. And these can be either, this is some categorizations of these, they can be septate, which means that there is a cell wall division between each cell of the hyphae, or they can be non-septate, or a cenocytic, which means that the entire cytoplasm is all connected, so all the cells aren't separated. They're kind of joined into this one large kind of structure here with all the nuclei. And then we also have this term called pseudohyphae, which is just whenever we have these budding, so these yeast or unicellular fungi that are budding, and they form these long chains of buds that kind of resemble hyphae, so we call them pseudohyphae as well, as you can see here. Okay, like I said, hyphae, they form tangled networks within soil, oftentimes called, they're in this collective all these branches are called mycelium. And then within these hyphae can actually interact with each other and undergo sexual reproduction. So you can imagine if you have this large root structure of mycelium, if some of the hyphae start mating and reproducing, then you might have a mushroom form and make this big reproductive structure here. And that's kind of how many mushrooms can arise from the soil, even though you can't see any structure above the ground. It's just all the mycelium is like kind of spread underground. And we'll look a little bit more closely at this process. And then finally, we have some fungi that are what we call dimorphic. So they can alternate between unicellular and multicellular depending on their environment, as well as many other factors. Just to remind you guys, fungi are eukaryotic, right? So they have nuclei, and they have what we call, remember, 80S ribosomes. So ribosomes are a little bit larger than prokaryotic 70S ribosomes. And then they kind of have all the standard eukaryotic cell structures that you think of when you think of human cells, such as like smooth, rough ER, Golgi, mitochondria, vacuoles. I guess vacuoles aren't necessarily in human cells, but you get the gist. So they have kind of all the standard things. There's and then there's a few things that are unique that we'll look at here. But before we do that, I have this like general diagram that shows all the components within this. This is a yeast cell. Like I said, it has all the standard things that an animal cell would have. A few exceptions are this, of course, the bud coming off, this new budding cell here. Then we also have what we call the septum, which is just when a new cell wall begins to form between the two cells that are separating here. And then we also have what we call a bud scar. And this is where there's kind of, it's literally a scar where the cytoplasm and the cell wall, you can kind of see where the cell started budding before. So of course there would have been a cell here that this cell budded off of. So when it's separated, it kind of leaves this scar called a bud scar. And we have the vacuole, which isn't always present in animal cells, although it is present in plant cells. And this vacuole is just used for storage of either nutrients, water, sometimes waste products, etc. So, and I would encourage you to go ahead and review all these standard organelles that you've already heard about on your own, just so that you don't get tripped up on some of the simple things, right? So like I've been mentioning, the fungi have a cell wall, and it's similar to the bacterial cell wall in terms of it being composed of sugar. So this time it's composed of what we call chitin, which is also found in insect exoskeletons, that insect shell on the back of like insect backs. And then it's a long chain of NAM and acetylglucosamine. If you remember, this was one of the components in the peptidoglycan bacterial cell wall. Peptidoglycan was peptidoglycan equals NAM plus NAG and acetylglutamic acid. Oh, sorry, I think I mixed this up here. This should be N-acetylglucosamine is NAG. That's my bad. NAG. Yeah. And then NAM is the other one in bacterial cell. Yeah. Also it has some other embedded molecules such as glucans, which are just sugar molecules, and then it also has some mannins/mannanoproteins. These are glycoproteins or sugars and proteins that are connected together, linked together, and these just help provide rigidity by linking the cell wall together, all these strands of NAM, and then they also provide rigidity and virulence here. Okay, next, the cell membrane. So fungi, they have one cell membrane that is within, quote unquote, under the cell wall, right? And it has kind of the same eukaryotic structure that we've looked at in animal cells, the phospholipid bilayer, right? The fluid mosaic model, all the embedded proteins. However, they contain, instead of cholesterol, like in our cells, they contain a similar sterol derivative called ergosterol. So that's one of the key differences, and this does the same thing as cholesterol. It regulates the fluidity of the cell membrane, so it makes sure that the membrane doesn't melt away, and also make sure that it doesn't freeze up and get too solid. And it has also been implicated in inducing an immune response when it's inside of a host like a human cell, or like a human, Okay, and then plasmids, briefly, just want to mention that yeast can also carry plasmids like bacteria. And in fact, artificial plasmids are very often used in the lab to run experiments on yeast and test the function of different genes. Okay, let's get into the sexual reproduction cycle of fungi. And this is multicellular fungi. So we have these hyphae in the diagram, right? What starts with two mycelia or two hyphae? Same idea. So basically, these hyphae, they have what we call mating types. So there's some of them are like plus, negative, and it depends on the species. Some fungi have like 50 different mating types or whatever. So this is just like, kind of just think of them as different types of cells that can interact together and mate. Whenever some of these mycelia come into contact, some of these mycelia will have haploid. So remember, haploid means one copy of the genome, one copy, whereas diploid is two copies. So some mycelia contain haploid gamete cells that are reproductive. So when these cells come into contact, when nearby hyphae come together, they can fuse and form a diploid cell. So the haploid, one copy plus one copy equals two copies of diploid cell. So this is like the same concept as like sperm plus egg equals zygote that has two copies of the chromosomes. So they'll fuse. First, the membranes will fuse together in what we call plasmagamy. This means like the cytoplasms are connected. Then the nuclei will kind of collide and fuse into each other and undergo what we call karyogamy. So now you have the diploid cell, and it'll start growing, developing, dividing, And what it does is often it'll form a large rigid structure, as you can see here. And this rigid structure, through meiosis, it'll get filled. Remember, meiosis is you start from one diploid, and then you get 4 haploid reproductive cells from reproductive cells. So through meiosis, this rigid structure will start to fill up with many, many, many haploid spores. And spores are just reproductive fungal cells. So what happens is you can see these structures here, right? These can, after they get full, they'll burst, and then they'll release up to tens of thousands of these haploid spores that can go, and then they'll plant themselves in the soil or somewhere far away, get blown away by the wind. Then they'll they'll plant themselves, and then they'll start multiplying, and they'll start growing into new mycelia. So this is how these fungi will spread. So of course, this is a very simplified view of the life cycle, and there's many different types, many alterations to this cycle based on what type of fungus we're looking at. But this is kind of a good general overview that is good for kind of umbrella knowledge here. And so this structure that I've been referencing can have many different names and morphologies depending on the type of the fungus. In general, we can call it a sporangium or a zygosporangium. But there's lots of diverse things. So for example, here we have what we call an ascus. So this is like a zygosporangium, except it's like a different type, and it's where we have like 8 spores within this linear structure, and then we have a bunch of them gathered here. And then another one is here where we have what we call basidia, and the spores kind of like hang off of these basidia on the bottom of these mushrooms. So the mushroom kind of ejects these spores outwards like this, downwards. Okay, so let's get into our only fungal organism on the microbes and agents list. This is Saccharomyces cerevisiae, or S. cerevisiae, and it's commonly known as either the brewer's yeast or the baker's yeast because it's very often used in bread production as well as alcohol production. And of course, it's a yeast, so it's unicellular. Although, cells can clump together, but here each individual cell is existing and metabolizing and, on its own. So it can perform what we call ethanol fermentation, which we'll get into in a future lecture. So this should have been covered in your bio class freshman year, but just as a review, it's when these yeast are depleted of oxygen, so they're in anoxic or anaerobic environments where they can't perform normal cellular respirations to get ATP. So what they'll do is they'll use this process called fermentation, where essentially you're producing less ATP, but you're also making ethanol and CO2. Ethanol is just alcohol. Ethanol and CO2 as a byproduct, and we can kind of harness these byproducts for our own food production or other purposes as well. So you can imagine if you cause fermentation and these yeast and stick them in like grape juice, then of course over time the yeast will produce wine from that. And then for CO2, right, we put the yeast into bread, then the CO2 gets produced and it makes the bread rise in the oven. And of course, we can briefly talk about the cell size. So they range from 5 to 10 micrometers or microns, meaning that they're a little bit bigger than most bacteria, but they're also smaller than human cells. Okay, looking at the life cycle of S. cerevisiae. So it has a unique life cycle that alternates between haploid and diploid forms. So again, I have this written down, but haploid is 1 copy, and then diploid is 2 genome copies. So for example, right, diploid would be like any of our normal cells, excluding our gamete cells. So when in the haploid stage, basically these cells can be divided into 2 types. Based on their gene expression, and they're called MAT-A and MAT-alpha, and these are think of them as male and female, essentially. So these haploid cells, they can undergo budding, like we looked at before, and they can keep making identical copies of themselves through asexual budding. So they can do that as much as they want, but if they can come into contact, then they can actually undergo sexual reproduction by fusing together and forming a diploid cell. Like I said, the analogy would be like a sperm and an egg, and then they come together and then make zygote. But in this instance, this quote-unquote sperm and the quote-unquote egg can exist on their own and divide on their own as well, for this analogy, essentially. So so here, both mating types, they naturally secrete, or they kind of pump out of the cell. They secrete what's called a pheromone, which is just a signaling. It's a short peptide, or a sequence of amino acids that they kind of constantly produce and shoot out of their cells. So MAT-A secretes what we call A factor, and then MAT-alpha secretes what we call alpha factor. And if one of the haploids senses the pheromone of the other mating type, so if a MAT-A cell senses alpha factor, then that'll activate this signaling pathway through these. So these guys have these signaling proteins. It'll activate that, and basically through a big cascade of signaling events, it'll cause the cells to what we call shmoo. And this is a very funny name that we'll get into the history of. So shmooing just means that this cell will start to elongate and make this kind of extension that'll go out and try to search for the other mating type. And as you can imagine, both of these guys will extend, and then they'll touch in the middle and then fuse like this. And you can see that under the microscope here, right? We add pheromone to these cells, and then the cells start shmooing, forming these elongations like this. And the shmoo actually comes from, it's some like random comic from like the 70s or something. I don't remember exactly, but there's just this character called a shmoo, and then the researchers that discovered this phenomenon thought that it looked kind of like this character, so they decided to name, this is the actual like literal scientific name, is called shmooing. It's very funny. Yeah, so once the cells shmoo and then fuse and then form a diploid cell with 2 copies of the genome, the cell again can reproduce asexually through budding in this repeated cycle over and over, however much it wants. But when we put the cell in a starvation condition, so low nutrient conditions, or what we call oligotrophic conditions, and usually this is when we deplete the cells of nitrogen. It'll undergo sporulation. So this sporulation process is somewhat similar to the endospores that we looked at in bacteria for the antibiotic lecture. But this time, the cell itself will undergo meiosis. So from one diploid cell, it'll produce 4 haploid spores, and then it'll create this spore hard shell structure around the 4 cells. And here, 2 of the spores will be type MAT-A, and then 2 of them will be MAT-alpha. So you kind of have a half-half division of the quote-unquote like male and female within the spore. And of course, after the environmental conditions come back to normal, then the spores will, what we call, germinate, and this shell will kind of go away, and then these haploid spores can again go out and then repeat the entire process and start from the beginning.